Question number 146. Select the divalent cation which increases the efficiency with which DNA enters the bacterium through pores in its cell wall. And the options given are phosphate ions, zinc ions, calcium ions, iron ions. And the correct answer to this question is calcium ions. Calcium ions are the divalent ions which are essential components to make the cells competent, and they increase the efficiency with which DNA enters the bacterium through pores in its cell wall. So the correct answer to this question is option number 3. Let's move on to the next question. Question number 147. A vectorless method for transferring the recombinant DNA into the host cell is, and the options given are biolistics, Agrobacterium tumefaciens, Cosmid, and BAC. And the correct answer to this question is option number 1, bialistic. Because Agrobacterium tumefaciens, Cosmid, and BAC are vector-mediated methods for transferring the recombinant DNA into the host cells, whereas plant cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA in a method called biolistics, and it doesn't require any vector. So the correct answer to this question is option number 1. Let's move on to the next question. Question number 148. Complete the analogy. Lysozyme is to bacteria, chitinase is to. And the options given are plant cell, fungus, animal cell, protozoan. And the correct answer to this question is option number 2, fungus. Chitinase is to fungus. Isolation of genetic material can be achieved by treating the cells with enzymes such as lysozyme by bacteria as given in the question, cellulase for plant cells, and chitinase for fungus. So option number 4, 3 and 1 are incorrect. And the correct answer to this question is option number 2. Let's proceed to the next question. Question number 149. In the given fragment of DNA, what will be the best choice of enzymes if only gene C is to be isolated without any flanking sequences? So you have to find out the best choice of enzymes among the given options. This is the sequence given and you have to actually take out only this gene. The very first option given is double digestion with PST1 and SAL1. So if you digest this DNA fragment with SAL1, which is represented by this, and PST1, it's going to cleave here and here and then here and here. So you may get this C sequence, but then you require 2 enzymes in it. The next option is single digestion with HIND3, and this cannot be a correct answer because HIND3 digestion will cut the sequence here. You will not get the entire C gene, or it may also cut at these sequences, thereby producing a C gene along with the flanking sequences. So this cannot be a correct answer. The next option is single digestion with PST1, and single digestion with PST1 produces C gene without any flanking sequence. So this is a correct answer to our question. And this is the best choice because only single enzyme is required to produce this gene. Whereas in the first option, as 2 enzymes are being used during this reaction, so this cannot be a correct answer. The last option is double digestion with HIND3 and SAL1 is also an incorrect option because it is not going to produce the C gene without any flanking sequence. So the correct answer to this question is option number 3. Let's move on to the next question. Question number 150. Ti plasmid of Agrobacterium tumefaciens is disarmed by, and the options given are insertional inactivation of beta-galactosidase gene. This is an incorrect option. Beta-galactosidase gene and antibiotic resistance gene are features of PUC8 plasmid and not Ti plasmid of Agrobacterium tumefaciens, because beta-galactosidase gene and antibiotic resistance genes are features of PUC8 plasmid and not of Ti plasmid of Agrobacterium tumefaciens. The next statement says insertion inactivation of antibiotic resistance gene. This is also incorrect. The next statement says replacing multiple cloning site. MCS is for multiple cloning sites from the Ti plasmid with new ori. This is also incorrect. And the last option says replacement of tDNA with our gene of interest. And as it is a correct option, this is a correct answer to our question. Because TI plasmid has its own origin of replication gene. TI plasmid has been disarmed by replacement of transfer DNA or tDNA with our gene of interest. So the correct answer to this question is option number 4. Let's move on to the next question.